M13 phage vector is another important vector which can be used in cloning. So another phage used as a cloning vector is the filamentous phage M13. Filamentous means long, thin, filament like. M13 is a filamentous coliphage with a genome composed of single stranded circular DNA. So इसका genome जो है single stranded DNA है लेकिन circular है. So here you can see this is M13 bacteriophage और इसके अंदर ये आप देख सकते हैं ये जो है ये single stranded DNA fragment है जो कि circular fragment है. Joachim Messing and his co-workers endowed the phage DNA with the same beta-galactosidase gene fragment and multiple cloning sites found in the PUC family of vectors. In fact, the M13 vectors were engineered first, then the useful cloning sites were simply transferred to the PUC plasmid. So ये जो M13 वेक्टर्स हैं ये पहले बने थे और बाद में इनकी जो MCS जो है यूजफुल क्लोनिंग इसको साइट्स जो हैं इनको ट्रांसफ़र किया गया था PUC प्लाज्मिड में. The DNA in the phage particle itself is a single stranded, but after infecting an E. coli cell, the DNA is converted to a double stranded replicative form. This double-stranded replicative form of the phage DNA is used for cloning. So this is this life cycle of M13 phage. So this is M13 phage. It enters the bacterial cell through this pilus, F pilus, and after entering, you can see just after entering the cell, it is single-stranded. But soon after that it becomes double stranded replicative form and then it starts replication and these new particles are formed which then emerge out of the cell. So this is important for cloning purpose. After it is cut with one or two restriction enzymes at its multiple cloning site, foreign DNA with compatible ends can be inserted. This recombinant DNA is then used to transform host cells, giving rise to progeny phages that bear single-stranded recombinant DNA. The phage particles containing phage DNA are secreted from the transformed cells and can be collected from the growth medium. वो growth medium में secrete हो जाते हैं वहाँ से फिर collect किए जा सकते हैं. The main factor is that the genome of this phage is a single-stranded DNA. So DNA fragments cloned into this vector can be recovered in single-stranded form. Single-stranded DNA can be an aid to site-directed mutagenesis by which we can introduce specific premeditated alterations into a gene which is called site-directed mutagenesis. The nature of these vectors makes them ideal for techniques such as chain termination sequencing and in vitro mutagenesis, since both methods require single-stranded DNA. So this is the M13 phage vector. You can see this is the multiple cloning site, this is origin of replication and this is antibiotic resistant site. So this is M13 phage vector.